Okay, so this is the e-lecture for Lab 5. We're mainly going to be taking a look at cells. We're also going to be seeing how osmosis works in the presence of solute. So one of the key things here is the similarities and differences between prokaryotic and eukaryotic. And probably the first and best example of this is how to design an antibiotic. So you need to design a drug that kills prokaryotic cells, the bacteria, and not the human eukaryotic cells. So some of the key differences is that prokaryotes, for example, they replicate their DNA in a unique way and with unique enzymes than eukaryotes do. And so we can target drugs that attack this DNA mechanism and it won't hurt us. They also have cell walls. So if you put a drug in that attacks a cell wall, it'll kill the prokaryotes and it'll leave our eukaryotes alone. Okay, so you can take a look at these Prelabs. Pause if you want. Yep. So this is a good little video. Shows the inner life of the cell. Come on. So what you're seeing right there is the fluid plasma membrane. We're just kind of starting off to the races. Inside. So right there is the cytoskeleton. See how it holds the plasma membrane kind of together and coordinates everything. This is more of a strong structure. This is an intermediate fiber or intermediate filament. It doesn't change much. It's very stiff. This is an actin filament. This can assemble and deassemble very quickly in this spiral because of the fibers. Next up. So proteins can fissure and separate when it needs to. These are microtubules. They assemble at one end, seal into this nice like hollow strong tube, and they can disassemble just as quickly. This is important for cells to move around. This is a motor protein binding and then unbinding, and then binding again after torquing and moving forward, and it's carrying this big vesicle of nutrients in the cell. It's a really cool look, kind of what's going on inside the cell. Right there's the nucleus, and right here the nucleus spits out mRNAs that have been encoded from DNA. They are ultimately attracted to ribosomes, which attach to them, and ultimately start translating protein right there. So that's the protein. It's a great video. We'll probably watch it lab too, so we can head back. Okay. So lab safety, there's some stain. It's not too big of a deal. The cover slips that we'll use, so these are the little glass slides that you cover the slide with. You cover whatever you're looking at. Those go in the sharps when you're done. Wash all the slides, we'll reuse them, except the slides that you used for your cheek cells, because those are technically human biohazard, so definitely throw away those ones, the whole thing. Okay, so the first thing we're going to do also is look at how water behaves in the presence of solute, in this case salt. So we'll have 2 potatoes. You'll weigh them beforehand. You'll add H2O to both, and you'll let it sit for 40 minutes. Well, you also, and then you'll add salt to the other one. So water, and then water and salt. You'll let those stand for 40 minutes. So actually part 7, you'll start immediately, and we'll make sure to remind you that. So what you'll see is the water will be drawn to the high concentration of solute outside of the potato. So you should see a difference in the weight. These are some good resources for taking a look at your microscope. Definitely at least read up on some stuff. The main functional things obviously are your ocular lens, your adjustable lenses, your light, here's your focus, and here's how you move your slide. That's a basic functional overview. There's obviously a lot more detail to things that you probably know because you might get tested on it. I'm not 100%. There's a good little online resources here. Good thing about these microscopes is you can actually estimate size. So one line in the microscope grid is 0.2 millimeters. So if I see something that is just like this line right here, that length, I know that it's 0.2 millimeters. If it's 2 lines, it's 0.4. So it's an easy way to approximate the size of cells, and we'll be able to see that applied when we see how much larger eukaryotic cells are than the bacterial cells, the prokaryotes. So prokaryotes are important. They are the most ancient form of life. They are the most independent form of life there is, and they've survived everything, all major extinctions, because they're durable as a kind of as a species, if you want to say it, even though there's thousands of different types of bacteria. Super important. Not only do we always think about how to treat them, say in medicine, but they serve as one of the major parts of our body as the microbiome. So, because our gut and our body is filled with specific types of bacteria that help us digest food, help our immune system sometimes. It's a very symbiotic relationship. So you'll also get to take a look at microorganisms from pond water. And I saw them in lab today, and they're floating around, and they're all alive in there, and they're having a pretty good time. They might not under the microscope, but it's all right. This is, for some of you, this will be one of your first experiences working with a live organism. And it's definitely your first try with the question, how comfortable are you with microorganisms? Or no, using organisms as a model. Because the obvious line is, humans are not used as organisms in lab. Then, is it ethical to use primates? Is it ethical to use mice, for example? So every researcher can kind of make their own mind about that. For example, I usually don't work with mice because I just don't care for it as much. But this will be a great experience to see what microscopic life is like. So it's one of my favorite things to do in an intro. So you'll be able to identify those. You can get out your phone and find little identification pages for these. You can sketch them. We'll also take a look at an aquatic plant. We'll be able to see kind of this rigid cell wall you see around here, and how the exchange of water is so free in these plant tissues. We'll be able to see water kind of jettisoning through. I think those are the chloroplasts. I'm not 100% sure. The interesting thing about chloroplasts and mitochondria is that they're alive independently. Our mitochondria in our cells, they move around freely. They're their own little organisms. And if you read about in your book about endosymbiotic theory, you'll definitely see why that's the explanation. We'll get to look at onions. Sometimes if you're lucky, there'll be cells that are dividing. So this nucleus right here will be splitting apart. That's always a good thing to find. It's very cool. And then we'll take a look at our own cells. And they look very much like this, just little sacs, and you got a big nucleus, and that's it. But it's definitely cool to see for the first time. After all this is done, we'll take a look back at part 7, and we're going to see that the side with the salt molecules and the water molecule that can cross. So the water can cross from the potato into the extracellular water, right? The salt can't go into the potato, though. So what's going to happen is that the water is going to go out, and it's going to follow the salt because it's going to try and dilute the salt levels to the inherent levels that it's at in the potato. But that's impossible because it's too highly concentrated. So, and don't worry too much about gravity. The interactions of osmosis are so minuscule and small that adhesive forces of water usually take care of that. Okay, and that should be good.